Great. Well, welcome everyone to today's webinar, Food Safety in the Era of Alternative Proteins, Challenges and Solutions. My name is Karolenko. I'm a Scientific Program Manager for Food Microbiology at the Institute for the Advancement of Food and Nutrition Sciences, or IAFNS. So just a little bit about IAFNS before we get started. We are a nonprofit organization focused on advancing food and nutrition science for public benefit. IAFNS connects tripartite membership from government, industry, and academia to support collaboration and catalyze knowledge. IAFNS funds actionable research conducted by external researchers who are selected through an open, competitive process, and all of our IAFNS-funded projects are viewable on our website. IAFNS advances food and nutrition science for public benefit by convening experts across sectors to engage in dialogue, and conducts its work by leveraging pooled funded combined with in-kind contributions from public and private sectors. Just a quick note on logistics before we get started. There's two different formats that you might see on your interface. So this is version number 1. Starting at the top, you'll notice an orange arrow. This arrow allows you to expand or collapse that control window on your screen. Once expanded, you can go ahead and type your comments or questions into that question box. This will allow you to participate in the dialogue, and only questions will be available to IAFNS staff and not other attendees. This is the other format that you might see on your screen. Questions can be typed into that thought bubble with the question mark. And again, this will be a way for you to interact with the speakers, and we'll have time for a Q&A after both speakers are able to go. All right, so with that, let's go ahead and jump into our program. We have 2 great speakers today. Each speaker will speak for about 30 minutes, and then we'll have time for a Q&A after both speakers have presented. First up, we have Dr. Kantha Shelke. She's a senior lecturer at Johns Hopkins University and founder and principal at Corvus Blue LLC, a food science and nutrition research firm. Her work focuses on new ingredients and technologies that aim to improve public health. Today, her presentation will be covering what we know now regarding food safety and alternative proteins. So with that, I'm going to go ahead and turn it over to you, Dr. Shelke.
Well, thank you, Caitlin, and hello, everyone. While alternative proteins are the rage today, and we've had notable achievements in our march towards greater transparency, cleaner and more sustainable products, and a means to ending food insecurity and global hunger, conspicuously absent is the public discussion about the safety and toxicology of the ingredients and the processes that go into various alternate protein products and also the finished products, so both ingredients and products. Now, plant-based proteins and the term alternate proteins for that matter is a market term with no legal definition, but it continues to be one of the biggest trends in the food manufacturing sector, not only in the US, but around the world. This emerging class of ingredients is serving as a springboard for innovation, investment, and a tumultuous market frenzy, while paving the way for success, failure, and possibly litigation against manufacturers of foods and beverages. Hopefully, those who are mindful can and will explore purposefully, respectfully, and with transparency, and will pave the way for success, not only for the plant-based, but also for cultured meat that the next speaker is going to address, and also precision fermentation and insect proteins. Disclosures. I'm not affiliated with, nor do I have any vested interest in any of the ingredients, products, or companies that have been mentioned in this presentation. I will not be sharing any confidential or proprietary information that's included here. Now, my firm is a contract food science and research firm that works at the intersection of science, nutrition, safety, and regulation for the development and market realization of ingredients that are honestly helpful. I'm also a judge for the $15 million X Prize international competition for developing alternate proteins to fish and chicken. So I get to see some of the latest and greatest of developments from around the world, and I learn from them and learn from some of the most forward thinkers and innovators. What you see in the banner below are my affiliation with regards to Johns Hopkins, Faculty Advisory Board of McGill University, Board of Trustees of the Sonoran University of Health Sciences, advisor to IAFNS, I mentioned X Prize. I'm a technical editor, contributing editor at Prepared Foods magazine. Ditto for PLMA magazine, which is PLMA Live. I'm an investigative reporter for CBS Chicago, which means it's not a good idea to go with me for dinner, especially in Chicago, because they immediately think I'm investigating them. I also serve on the advisory board of Nutrition Business Journal and a small non-profit that I helped start called Midwest Food Bowls Alliance. So meatless eggs and meatless meats and eggs without chickens are doing away with the animal source, which means really requiring a sound fundamental in protein chemistry, toxicology, safety of these introduced proteins, because they are new, the extent of processing and natural state, nutritional equivalency, and food safety. This is very important. Proteins are one of the 3 major nutrient types that we ingest, not just humans, but also animals. The safety assessment of proteins in determining whether they have the potential to exert adverse effects is very important. This means going beyond allergenicity, as multiple comprehensive reviews have covered this topic, and researching for what really matters more. So the questions are, as a scientist, we should be asking continuously, are alternative proteins better? Are they safer? Are they more affordable? And are they truly more nutritious? We should also, at the same time, keep an eye on what do we know? What should we know? How do we confirm that what we have is right and what we think we know? And what about unintended consequences? What could they be? And how do you prevent them if they are detrimental? Now, the alternative protein scene has become quite attractive to investors and to enterprises in the protein-based sector. While they may be led by P's for protein and P's also for profit, they must be mindful that P is also for process, purity and proof, potential risk, and purpose, the underlying reason for that choice. So alternative proteins are a nascent category. The plant-based meat represents just a little over 1% of the meat industry in the US, and cultivated meat has not even hit the global market, especially in the US. But the investment in alternative proteins continues to grow, largely from global meat, dairy, and seafood enterprises. They are all keeping an eye on what's yet to come and making sure they're not missing anything. So what's driving alternative proteins? The number one reason is the growing scarcity of animal proteins, also the environmental implications. Traditional animal protein production is still believed to be intrinsically inefficient. Health concerns that are associated with animal proteins, animal welfare issues that's associated with animal proteins, and then you've got meat-borne infections like the Creutzfeldt-Jakob disease. So this is a rapidly progressive, invariably fatal neurodegenerative disorder that is believed to be caused by an abnormal isoform of a cellular glycoprotein known as the prion protein. So we are very concerned about that, and the concern also extends to other products that are being developed as analogs to meat protein. And then the growing belief of the health benefits of a protein-rich diet, especially in the US. Everybody here absolutely believes that it's protein, protein, protein. That's what you take when you get up, and that's the last thing you should take when you go to bed. But if you're going to talk about proteins, it's very important to know the fundamentals of protein. So there are 3 important things to keep in mind. One of them is quality. Quality matters. The FDA, a complete protein contains all of the essential amino acids in adequate amounts, because incomplete proteins contain inadequate amounts, and they may be missing 1 or more of the essential amino acids. Quantity matters. Protein quantity also relates to bioavailability, and digestibility. The PDCAS, which is a protein digestibility corrected amino acid score, is becoming increasingly important as people try to define what their product contains and the implications of their product in terms of nutrition. The DIAAS, which is the digestible indispensable amino acid score, is also in the works for regulatory enterprises. For matters, the intactness of extracted and isolated proteins actually affects functionality and physiology. So the way the protein was laid in nature, in the legume, grain, nut, or whatever agricultural crop, is a very important matter because it's connected to and has implications to allergenicity and safety. So when you're extracting proteins and you're isolating them, how do we know that that intactness has been maintained and that what we've got is essentially intact protein? Because our immune system does one thing. It looks out for unusual configurations of foreign proteins. And when we start messing around with the configuration, that's when we start developing different types of sensitivities because our immune system does not recognize them. Structure and function matter. Now, animal proteins are in no way the same as plant proteins. There is distinct difference. Animal proteins have a structural role, they're complete, and nutritionally speaking, they're digestible. They have off notes only when they're spoiled, and they're very familiar to Western consumers. Plant proteins, on the other hand, are mostly functional. They're incomplete unless they're balanced, as in the case of soy. They're less digestible. They come with some digestive issues. They have a wide range of off notes, particularly if they're not treated in the right way. And people in the Western market are very unfamiliar with the various flavors, etc., of plant proteins. Now, the schematic that you see here on your, I believe our image has been switched, so I think it's on the left-hand side. So the schematic that you see, it shows the simple steps that most people believe that their plant protein goes through to land in their hands. That would be the case if the plant-based protein ingredient or the finished product was merely a matter of simple processes to convert the naturally occurring plant and the crop source of the protein into a finished product. So if you were taking bread and you were taking wheat and converting that into a loaf of bread, or if you were taking soy, grinding it up and making a soy-milk out of it, that's the process you go through. In reality, the other two schematics that you see, these are what protein concentrates and isolates typically go through in order to yield the level of protein, the functionality that's expected to create the plant-based analog of traditional animal-based foods. Now, concentrates are popular because they're less expensive, while isolates are more expensive and are still not as prevalent in the food product sector. But when you're looking at this aspect of alternate proteins, the isolates, the concentrates, or the extracted proteins, then you have to think about sustainability, the source, the waste, the spoilage. If it is a clean label product, there is no pass on ingredient type and name. Food safety, have you substantiated? And how have you made sure that it's not contaminated? Also, of course, is commercial viability and affordability, and most importantly, unintended consequences. Definitions matter. Plant-based generally refers to mostly products that are mostly solely made of vegetables, grains, pulses, or foods, and they are derived from plants, and they do not contain animal products. Now, cell-cultured meat, which is another alternate protein, is an animal-based protein food that is made by cultivating animal cells, and it eliminates raising and farming animals for food. Precision fermentation is another one. Cell factories of customized microbes and microbial hosts are used to produce functional ingredients like casein and other different kinds of proteins that were once rather wastefully derived from animals and plants. And then a new entry into our marketplace, but definitely has been in use in other parts of the world, is insect-based foods. So these are food products which use insect-derived ingredients in the preparation. But what do consumers think of plant-based proteins? So consumers perceive alternative proteins as healthier. They believe they are friendlier to the environment. People want convenient, affordable, and tasty food products with alternative proteins, but they've also wanted to do no harm. Now, the policy context and market trends reveal increasing business opportunities, and consumer beliefs, perception, and understanding are all evolving and must continue to evolve. So having a very simplistic view of plant-based or alternative proteins will not do. And most importantly, innovation is needed to ensure honestly healthy and safe alternative proteins. So when someone looks at, for example, this plant lettuce tomato or a plant-based protein hamburger, this is what they see. But if you were to show this image and ask them if they would eat a food ingredient that came out of this food production facility, 9 times out of 10, the answer would be no. Fact of the matter is, this particular image shows how the production facility for leg hemoglobin, which is a soy derivative that is used to color and to make plant-based proteins appear more like its meat counterpart. So what you see over here are 3 different hamburgers with distinctly different implications on health and environment. The objective here is to show that although all 3 might be considered a hamburger, because they are sold in the market as hamburger, that's what people expect when they bite into them. They're expecting the taste, the texture, and they're also expecting to a great amount the physiological implications to be the same across. But the truth is, although these 3 products are sold as hamburger, they come under different regulatory jurisdictions. They're all innovations of their time. So what you see is a plant-based, a cell-based, and then being possible burger, and McDonald's, McDonald's hamburger sandwich. But their regulatory, physiological, safety, and environmental implications are all distinctly different. This shows that although these alternative proteins are being used to make products that look like, feel like, taste like, and are supposed to behave like their meat or animal-derived counterparts, that's not always the case. So biology is complex and often context-dependent. Food is a part of biology, and every food, even plant-based, is a molecule of some kind. So the interest in plant-based has usually been because they believe that it is more natural, and for some reason they think that it contains no chemicals. I chose the word molecule instead to make it less harsh. But the key challenges are the same across the board, whether it's for plant-based, cell-based, or insect-based. It includes nutritional equivalence, taste, texture, visual appeal, and most of all, safety. So when you look at nutritional implications, when a person sees the Impossible Burger or Beyond Meat, just picking on 2 brands that are well known, what they are expecting are the nutritional implications of a meat-based hamburger. But the enjoyment and the satisfaction that they want is really what they are perceiving that's on the left-hand side. Completely made of plants, benign, and it's really good for you. So the implications to be aware of are the long-term effects on metabolic health, the physiology of intact plant matrices versus extracts and isolates, as I've shown you before, the effect on the gut microbiome, because they're distinctly different, and are they really a solution to complex problems in food insecurity as it is touted to be so? Then there is this matter of allergens and allergenicity, which includes allergen prevalence and potency. Currently, there is no clear guidance to determine acceptance or rejection of a new protein or an alternative protein in a food product based on potential allergenicity. While there are low levels of dietary exposure to functionally intact introduced proteins, the threshold of toxicological concern, the TTC concept, could be used to assess thresholds of exposure that have been shown to have very large margins of safety, which would preclude the need for toxicity studies. This refers to crops with functionally intact introduced proteins, to say enhancing the gluten content or the protein content of a particular crop. But there's something else, and that is the sensitivity that tends to build over time. Take grain millers, for example. My formal education was as a cereal chemist, and what we noticed was grain millers would gradually develop sensitivity and even allergic reactions to the grains that they've been handling for years. I mean, that's their career. They've been doing this for decades. But because of the extended exposure to an allergen, they have started to become sensitive or actually even allergic. So this begs the question, will we develop some form of sensitivity to prolonged exposure to these isolates and concentrates that our physiologies have not quite yet kept up? Then the other part of it is that there have been no reports of direct carcinogenic, teratogenic, or mutagenic effects associated with the ingestion of alternative proteins in general. Food safety scientists in regulatory agencies have not considered it necessary to conduct chronic toxicology testing of proteins, whether they come from GM sources or not. And these proteins are not considered to be toxic based on their known biochemical function. The intact proteins and the potential peptide degradation products are also not similar to known protein or peptide toxins as determined by bioinformatics. And so the residual levels of introduced proteins are present at very low levels in food products, and we are capable of ingesting them and have a low potential for systematic absorption of the intact protein is pretty negligible. Now, chronic testing is not generally needed to identify the potential toxicity of known protein toxins, as this is usually manifest after acute or short-term dietary exposure based on the known biochemical functions of protein toxins. The extent of processing and the natural state of proteins also matters. Contaminants and residues from protein sources and the extraction method really plays a very important role here. So the method of processing, extrusion, high temperature, high pressure, anything else that you would use, they can affect the form and texture. They can reduce microbes, they can help extend shelf life, but they may also have a potential for producing carcinogens, depending on what else is added to that particular food ingredient. There is a risk also of microbial contamination from post-processing handling. So helping consumers understand how to handle these products is very important, and consumer education is something that is really something that this industry should be looking at. What is important is that the alternative proteins may look like and taste like, but is not what it appears to be. And that is a very important part of this whole experience. Complications. So when you're looking at formulation and technical realities, there are a ton of complications because there are no industry standards. A protein extract from one supplier could be drastically different from another. And keep in mind, many of the suppliers in the alternative protein sector are novices and newcomers, and they've gotten into this business without having the fundamental science, experience, and knowledge. The other part of it is that these sources come with unique challenges and varying spoilage mechanisms. So when when a hamburger or a milk, dairy milk, gets spoiled, we have years of experience. We know exactly the mechanism of spoilage, what microbes would be involved, and what the consequences could be. But when you're talking about formulating with a range of protein alternatives, then you are dealing with some very unique challenges and getting into some unknown territory. And there's this unintended consequences of ingredient choices, because some of us believe that if something is good for us, more of it is better. And the amount that was consumed of these alternative proteins is in an unprecedented way. So our human physiology has not been used to consuming so much isolated, extracted proteins, because we were always consuming it in the intact, original, native form. Then there's the concept of and the implications of anti-nutrients, interactions with our ingredients, off flavors. Are those off flavors just because of what the inherent, because of the inherent molecules and the alternative protein, or are those off flavors because something has gone awry? And then the quality implications. Now, plant-based proteins and alternative proteins are not really new or complicated in some ways. It's mainly a matter of rearranging and reapportioning the basic materials from which both plants and animals are made. So it's the fats, proteins, carbohydrates, they've been turned around, and you're making some new products. Asian Buddhists have been kneading flour and water for centuries, washing away the starch and making mock meat from wheat, gluten, for example. Tofu and fermented tofu has been made for more than 1,500 years. So this has always been in the market use. What is new is that this sector, which was once fostered by hippies and animal rights organizations and some religious groups that practice vegetarianism and veganism, is now moving away from that obscure corner of the grocery store, right next to the day-old bread and gross produce, but it's now moving into mainstream aisles and flashily. So people are now buying them in tons, and they're consuming them copious amounts. So that is what could cause an issue, because what is new is the setting and how consumer lifestyles and demands are pushing the envelope of science for proteins that are made with the core plant-based protein or alternative protein. The functional ingredients, they now have to look and feel and have the properties of their animal-based counterpart. So they need some alternative proteins. Added ingredients so that they are at par with nutrition. The fat and carbohydrates that are added to mimic the taste and the texture of the plant-based original. So this is the original plant-based products did not always have this level or these kinds of fats and carbohydrates. Needless to say that physiological effects are significantly different. Then there's this thing about processing aids. So are they really needed? How do they hold up during processing and handling? And do they need special ingredients to serve as processing aids? There's also concern about preservatives. Preservatives, because our foods today, whether they are alternative proteins, plant-based or not, they traverse thousands of miles in a wide range of environmental conditions before they land in the hands of the consumer. And so but they have an expectation of quality and safety as if it was freshly prepared right across the aisle. So that makes a difference, the number of ingredients and ingredient interactions. Then comes the concern about product development needs. So for a product developer or a formulator, functionality matters. Can this alternative protein replace animal fats? Can it mimic traditional texture with flavor experiences and make it succulent? Because that's usually the concern. What about nutrition and enrichment and micronutrients? What about the source, how it's processed? Sustainability claims and can be substantiated. The cost, the availability, and consistency as the demand grows up. And most of all, resonance with consumer expectations, along with proof of safety, particularly when you're talking about post-production products. Then comes, of course, the microbial issues. Plant alternatives tend to have a near neutral pH, they're high in protein, and they have a high moisture content. All of these are the perfect storm for an increased susceptibility to microbial growth. They have made by people who often have a shallow knowledge of plant-based categories and the underlying science, both in terms of product development and in safety. Also, what you have to keep in mind are the different levels and variety of microbes, because you are now talking about a product that may contain 2 or more alternatives that are agricultural crop-based that come from very different parts of the world and have very different categories of the types of microbes associated with them. And you could also have heat-resistant spores and heat-resistant toxins, something to keep in mind. Regulatory matters is also very important. Alternative proteins and plant proteins are not defined by the FDA, USDA, or any other global regulatory agency. So it's unwise to extrapolate food safety rationale from the regulations that were really made for animal-based to plant-based. Dairy, here we're talking about milk, egg, poultry, meat, seafood. They all have a consistent nutritional profile. They have physical characteristics that consumers are very well aware of. Their microbiology is very well documented. So is the history of use and industry knowledge. We know when things go wrong, what has gone wrong, and how to fix it. When you talk about plant sources, you're talking about unique macro components that are susceptible to microbes that we may not always be familiar with, and a different mode of spoilage. Processing conditions matter. The heat, pressure, pH, solvents, all of these can change protein properties, how they react with other ingredients, and their susceptibility to microbial contamination, both at the starting point and the growth pattern and post-handling contamination. So regulatory agencies are only now just beginning to gain expertise in this, because this requires a very unique expertise and a new kind of expertise. So the potential for improvement across the alternative protein chain is a very important one, because protein alternatives are not necessarily a healthy choice for everyone. It's very different. The source selection matters. Ingredient handling matters. The types of mechanical and chemical processes to fine-tune the functionality, but most of all to enhance safety. Formulation and manufacturing matters. Matching the ingredient and the processes to deliver on taste, texture, aroma, and structure without compromising safety is of utmost importance. Of course, if you're in business, you want to optimize the source material that they use in different breeding technologies or processing technologies. Also food science. It's a food science-based process and focus on process and outcome versus philosophy-based, which is really how these products came to be. So now you're looking at and saying consumers want it, the market demands it. How can we apply food science to it to make sure it delivers on expectations and is also safe with it? So this is a call to the industry and call to various disciplines to collaborate: agronomy, biochem, cell biology, bioprocess engineering, food science, genetics, and stem cell biology. And very important caution: vigilance is critical, because you must be prepared for unintended consequences of alternate proteins. Now, food is socially very sensitive, and food manufacturers, and especially those who are making plant-based proteins and finished products, are using these plant proteins in the very same manner as they would use animal-based products. But the important part of it is the lack of transparency, because the food industry is very much like, that one relative we all have who will not share their recipe for the brownie or whatever, just so our product doesn't, match, is not as good as hers. But this lack of transparency is a different matter when you're talking about food safety in the food industry, because lack of transparency increases speculation. And if that speculation is coming along with associated inaccuracy, it is bound to foster distrust. They say the path to hell is paved with the best of intentions, but the road to good intentions is paved with plaintiff lawyers. So be prepared that although alternative proteins is a growing market, it is also increasingly attractive to those who will be, suing for one reason or the other, because we have to make sure that what we deliver is what we promise is indeed what we deliver. So the future of plant proteins is not just in the hands of consumers, but also regulatory and policymakers. Transparency and integrity are crucial in ensuring that these foods are safe for people and for the planet. For only the honestly helpful shall succeed. Thank you.
Excellent. Thank you, Dr. Shelke. All right, we're going to go ahead and move on to our next presenter. Her name is Dr. Bree Duffy. She's Director of Responsible Research and Innovation in the U.S. at New Harvest, a nonprofit research institute that supports open and public cultured meat research. With a background in tissue engineering and a passion for a transition to cellular agriculture, Dr. Duffy works to build a more sustainable and equitable food system for all. And she'll be giving a talk more focused on cell-based foods of alternative proteins. So with that, Dr. Duffy, I'm going to pass it over to you.
Thank you so much, Caitlin. And thank you, Dr. Shelke. You brought up so many great points. So as Caitlin mentioned, I am Brianna Duffy, and I'm the U.S. Director of Responsible Research and Innovation at New Harvest. And I'm going to be focusing specifically on cell-based foods. Thank you so much, Dr. Shelke, for kind of the background and all of that. And I absolutely agree with a lot of your points for option and lack of transparency. You'll see those themes highlighted in mine as well. So I'm going to be giving some perspectives on food safety of cell-based foods, both in North America and globally. So first, for a little bit of background on the organization that I'm coming from. So New Harvest is a global donor-funded nonprofit. We were established in 2003, back when the idea of cellular agriculture, cell-based foods was like purely sci-fi. Nothing had names or any companies out there. So we've really been around for quite a while, and the trajectory of the field is incredible to watch. So we are based in the US, Canada, and the Netherlands, but our work really is global. So our mission is to maximize the positive impacts of cellular agriculture. And this was actually a very intentionally shifted mission back in 2020. We changed our mission from building the field of cellular agriculture to maximizing its positive impacts. And that was really based around this idea that the trajectory of technology is not a given. Again, you have unintended consequences. And if we want to see the positive benefits that this technology has the potential, we really need to work on shepherding the technology in that direction. So we work to support groundbreaking open research, really with the goal of guiding the development and governance of this field. To do that, we really try to break down the silos between disciplines and sectors, because this field really does bring together a lot of different disciplines that may be mature in their own sectors, but are now needing to interface. And we also provide leading expertise on identifying and addressing neglected issues in this field. So what is cellular agriculture or cell-based foods? So these are both umbrella terms that are used for this idea of making food from cells rather than whole organisms. And this can include a whole variety of potential food products. And it also includes a couple of different technologies that I just wanted to overview so everyone was on the same page. I kind of put them in two buckets of products that are made by cells. And so this would be things like precision fermentation or cultured proteins versus products that are made of cells. That would be things like cultured meat and seafood. So to start off with the arguably slightly simpler one, products made by cells. If you're talking about something like precision fermentation, this is going to be the microbial cell culture production of single molecules. So things like milk proteins, egg proteins, or flavors and ingredients that might be used in other products. And this process has been used for quite a while to produce things. And in food, even rennet, an enzyme used in cheese making, has been made this way. It's primarily today in the market, and that's been that way since the FDA approved it back in the 1990s. Rennet used to come from the stomach lining of cats. So it's kind of an example of that transition to these products being made through this technology. But its application to things like milk and eggs is really quite novel. We've only seen that in the last decade. So there's a lot of work being done here. Also, a really interesting technology that fits in this bucket is actually mammalian cell culture for production of these proteins. So we're seeing this on the market in things like milk or collagens. This figure comes from a company, Biomilk, that's using mammary epithelial cells to produce breast milk. And so the idea here is that they can provide the kind of complexity of molecules that you'd be looking for in these products, but rather than precision fermentation, that it's going to be very individual. Now, moving on to products made of cells, this is what most of my talk is going to be focused on, both because I think it's a bit more complex from my technology and regulatory standpoint, and it's also my own expertise. So primarily I'll be focusing on this. So the technique here is tissue engineering, which is being derived from decades of research in the medical field. So really translating that work from the medical field into food comes with its own challenges and unique constraints. So the way that this works is you're going to take cells from an animal, grow those in a laboratory, eventually up into a manufacturing facility, probably growing them in these large bioreactor tanks, which would look quite similar to a brewery. And then in the end product, you're going to get some meat product, and that can be called cultured, or some people call it cultivated, or cell-based, or cell cultured. There's a lot of names for this right now, meat or seafood. And you can make other products like leather. There's companies making chocolate. A lot of opportunities here, but I'm going to focus specifically on the meat and seafood. So just to go into a little more detail about the process for these products and then the potential food safety hazards that can come from them. And this is by no means an exhaustive list, but just kind of highlighting some of the ones that I think are of particular interest. So first, you have the cell sourcing from an animal. And of course, there you have potential animal source pathogens that need to be looked at. And there's a high likelihood of antibiotic use at this point to combat that, although antibiotics are a food safety hazard themselves. You also have the potential for veterinary drug residues and other things that can transfer from that host animal. Then those cells are going to undergo probably cell line development to optimize them for the remaining process in the manufacturing facility. And so that could include things like genetic modification. It does not need to, but that is certainly a potential option when developing these cell lines to ensure they're optimized for this production process. So then you're going to have the production process itself, which is going to include the bioreactors, which the cells are grown in, the culture medium, which is the liquid that the cells are grown in, and that's really going to provide all of the nutrients and oxygen signaling molecules you normally get in the body. And then structural materials, which most of these cells require a surface to grow on. They can't just grow floating in the culture medium. And so pictured here is a microkaryote, a tiny bead that the cells actually grow around. That's commonly used at the proliferation phase, which is where you're just trying to grow as much of these cells as you can, really grow out your population. Then you transition into a tissue formation or maturation phase where you're actually forming that muscle tissue that you're looking for. And there you might use a structural material that's a little bit larger, looks more similar to the structure of a steak or whatever you're trying to make. So in each of these, there are some unique food safety hazards that could come in. Pathogens really are a possibility in all of these stages. You might need to look at the contact with metals where you're in that production phase. In culture media, you're using a lot of different ingredients, some of which may have a history of safety use in food, but not always. And so you need to look at the residues of those ingredients. Are they accumulating during this process, or are they detonating out? You need to look at allergens of those ingredients, as well as for the structural materials that you're using. And then for structural materials, there's kind of two approaches. You could have a material that you're intending to be in the end product. It might also be providing some sort of taste or texture to the end product. Or you may have materials you're planning to remove or will be degraded during the process. And so looking at those residues and treating those different classes of structural materials differently. And then finally, you have your harvesting and food processing, which may look quite similar to a lot of other types of food processing, a lot of the same types of risks here. But it's interesting to think about where there may be differences. So a good example is the physiochemical transformations. You see this in food processing for other products as well, but you might have novel ingredients in these products. So you need to look at the unique physicochemical transformations that could occur from those products. So as you kind of saw throughout that brief overview, there are a lot of potential food safety hazards that kind of span the whole process. I think microbial contamination is a really interesting one to look at. That really can occur at any stage. And in a lot of ways, there are relevant standard control measures that we can pull from a lot of other industries early on in the process. It might be that we're pulling those from pharmaceutical industries that do a similar process of growing cells in large bioreactors. At later processes, we may be looking at for analogies from traditional food processing. So this is everything from sterilization of the ingredients and equipment used. Antimicrobials can be used early on, although those are also themselves a potential hazard. So trying to only use them early on, and the intent that they're then moved away by the end, or animal health inspections and certifications when you're sourcing your cells. So a lot of these are really can be drawn from other industries. But again, you have to look at the differences in these products from existing products. So in microbial contamination, when at the food processing stage, it may look quite similar to typical products, but the innate microbiome of these meat products compared to meats that are derived from animal slaughter is likely to be quite different. A lot of companies like to tout that as a benefit, which it is, that you're likely to have a much cleaner product, but that innate microbiome actually has an impact on the ability for pathogenic bacteria to come in and colonize. So how is that going to impact the potential for food safety hazards during processing? Or is the nutrient content of these products going to be different, such that the growth of potential pathogens on the product during food processing might be different? So again, like, these questions that need to be asked because these products are a bit different. So switching gears a little bit to talk about the state of the industry. As Dr. Shelke mentioned, it has really exploded recently. So just a decade ago, there were a small number of companies, the first demonstration of a product. And today we're seeing over 200 companies globally with over $6 billion invested. It's just grown enormously in the last decade. Bringing it to the North American context, there's about 100 companies. So about half of the companies globally are based in the US and Canada. And I need to update this map to include Canada as well, but at least in the US, you can see it's mostly in California and primarily on the coast. And there's about 50 academic groups doing research in this space. I've just highlighted the ones here that are kind of the main hubs, have the most work going on. And the 3 with stars are the ones that, as far as I'm aware of, are doing safety research, although it's quite early. And there have been 2 regulatory approvals for cultured meat products. So those 2 approvals were done this year for the companies Good Meat and Outside Foods. And the approval process in the US is quite interesting. So I thought I would do a bit of an overview of that. So foods in the US are regulated by both the FDA and the USDA. And in the case of these products, the type of processes could fall under both jurisdictions. So there's been an agreement that it will be jointly regulated by the two agencies. So early on in the production process, when we're looking at cell collection, cell growth, and differentiation, that is overseen by the Food and Drug Administration. Then at the point of harvest, you see a transfer of regulatory oversight to the USDA, which oversees the processing, labeling, packaging, and the final product. And that's really because that's what they do for typical meat products as well. When you have animal slaughter, they oversee those processes. So it's really a separation of labor based on experience. An exception to this is game meat and toasty foods, which are fully regulated by the FDA, and that's because they are fully regulated by the FDA in conventional production as well. And I know I said I would mostly focus on cultured meat, but just to go back to precision fermentation for a moment, those are regulated through FDA GROS, or generally recognized as safe process. So it is much simpler, only done by the FDA, and we do have a couple of products in supermarkets now. So with approvals finally a reality in the US, how does that kind of change the landscape? And those are just some thoughts that I think are important for the industry to keep in mind. So, I mean, the US is a major region, so it's really important that we set a responsible standard as we move forward, both in food safety and also how the technology is implemented into society, which I know is not the topic of this stuff, but is something you'll hear space about a lot. Also, as we see these approvals coming out, the hype is only getting stronger, both positive and negative hype. And I think it's really important that we try to ground that in reality, understand the actual state of the technology. So neither of these approvals are at the scale of production, at cost parity, or with the sustainability benchmarks that we're looking for in these products. So they're incredibly exciting. It's a great moment for or step forward for the industry, but we need to keep that into perspective that there's a lot to be done still that's going to continue to need regulatory approval as the processes change. Also, skepticism naturally arises in the absence of independent data, and that is the state we're at now. So that's really a challenge as we have this hype versus reality question, is do we have the independent data in order to address some of the skepticism that's rising? And then finally, where are we actually at on safety as a whole? So I would argue that we're actually still at a very early stage. So I'm going to talk a little bit more about that. So where are we at on food safety? Well, we have these two regulatory examples, a few more if you include Singapore, but all of these are for really early stage ground chicken products. So that's just a very small part of the market. Market doesn't exist yet, part of the technology space. So there's still a lot to be done in other categories of products. Currently, regulators take a case-by-case approach to reviewing these products. So they generally accept any data that's generated using a range of methodologies, as long as the developers show they're scientifically valid. Not really because there are no industry-specific standards or validated methodologies for these technologies. And as we've talked to regulators, they understand that setting those now, while it accelerate their review process, would potentially hinder innovation. So there's this kind of interesting back and forth between wanting to spur innovation, but not needing to regulate. There is really limited available data on these products. And so the two FDA reviews did put out some information from the safety dossiers from the companies, and that's pretty much all of the data that we have at the moment. We have some data from research labs, but that's pretty preliminary and academic, which is incredibly important, but there's very limited data on the actual products. And there's also very limited safety research occurring outside of industry. So I did want to highlight this recent report that was put out by the UN Food and Agriculture Organization, because it is a really great step into safety research in this space. So part of the report goes through an extensive food safety hazard identification, going through an extensive list of sources of food safety hazards, potential mitigation and testing and control measures, and relevant barriers, as I was saying, from other industries. And so this is really helpful resource as a starting point, but it's just the starting point. FAO even knows there's a lot of work to be done. And in order to do that work, they cite the need for more data and research to assess these potential hazards, and more data sharing on a global and intergovernmental level if we want to create an atmosphere of openness and trust around this technology. So as part of this talk, I was asked this question of how do we build a safe cellular agriculture system? And I think ecosystem, and I think that's a really great question. And I would say we do have to go back to the tenets of responsible research and innovation. So that includes diversity and inclusion, including all actors that have research and innovation are conducted, anticipation and reflection on the implications of technology, which I think is especially relevant when we're talking about safety, openness and transparency, which is really an initiative for establishing trust both amongst the industry and regulators, but also consumers, responsiveness and adaptation to change, which is also very important when we're looking at society. So in the context of how we actually do this in the context of safety right now in the industry, I think what we need is more proactive safety research. And we need to build public-private partnerships to foster that collaboration across all of the actors. And we need to make more of that data open. And this is especially challenging to do this in this industry for a few reasons, and one of which is the way that it actually evolves. So a classic biotech pathway usually sees significant public funding upfront for early stage research at academic institutions, and then you see translation of that research to private funding for product demonstration and scale up. What we're seeing in this industry, or have seen in the last decade, is far more private funding than public funding. So a lot of that early stage research is actually happening at private companies, in addition to the product demonstration and scale up. And so what that means is that we're actually seeing a lot of that research happening behind IP creation. We're seeing siloing of the work happening and duplication. That's especially true around safety, where each individual company is doing its own safety methods, development, and data generation kind of in secret. And so this has led to a lot of excitement, but also really holding back the industry as far as moving forward on safety research. So what we need more of is more pre-competitive research that can lead to transparent data, data sharing and collaboration to really increase and accelerate this type of work across the entire industry. And so I wanted to share one of the things that New Harvest is doing on this, which is a culture and meat safety initiative, an initiative between Harvest and Virgo Advisors, which is an independent safety consulting firm. And this is an industry-wide initiative to address this shared challenge of a lack of data to demonstrate safety of culture meat. And so this began in 2020 with engagement with the industry, where we brought together 50 companies who, through a series of one-on-one engagements and virtual workshops, shared their manufacturing process to identify potential safety hazards that were reviewed in this publication. But what I really want to talk about today was the second phase of this work, which actually just wrapped up, where we actually worked with governmental scientists and regulators. So we convened 48 of these participants from 15 jurisdictions around the world, again, through a series of one-on-one engagements and workshops. And that was recently published in this paper showing their perspectives on these research priorities. So the goal of this phase was really to identify the research priorities from those who are actually going to be performing the safety assessment. And so you can see here, so it's a large figure, but kind of an overview of the research Lifespaces that were identified by these participants. And so this includes research that's needed to understand the process, because these all are novel processes. And next, research to understand the product, because as Shelke was saying, is although these products may be marketed as the same, they have a lot of differences. There are a lot of implications of these processes that need to be evaluated. And then we need research to develop the methods and safety approaches necessary to evaluate these and kind of work towards some standards and validated methods for the industry. And underlying all of this, we heard from across the board of these regulators we spoke to this need for more publicly available databases and methods to develop safety testing. But just zooming in on one of these as an example, so the inputs of these processes are quite different from the inputs you would see in a conventional meat production. And so we need to identify and characterize all of the potential inputs that we can see. So this is everything from cultured media to structural materials to the cell lines being used, anything going into the process. And from those inputs, there is a lot of research needed to understand the product. And so we're, for example, looking at the residues of these inputs in the final product. We need to measure the residue levels, as well as evaluate whether or not these inputs are accumulating or are they diluting out. The research needed there is obviously to develop approaches for residue risk assessment. And an interesting one, which the second time this is coming up today, is the TTC. I mean, they've cited that this is such a valuable tool for assessing these types of products, but it currently doesn't include a lot of the classes of substances we're interested in here, for example, bioactive molecules. So an expansion of the TTC to better address some of these other types of inputs would be beneficial. Or, for example, the composition question was a really big one that came up. What are the conventional parameters that we're comparing to when we look at the composition of these products versus conventional products? Does it matter whether the composition are the same? And if it's not, how do we communicate that effectively? And this is, again, all in the perspective of regulatory review. Obviously, composition is very important for lots of reasons, but from a regulatory perspective, there are a lot of questions about how it should be taken into account. So going back to that question of how do we build safe cellular agriculture ecosystems, I would argue we really need this proactive safety research to anticipate the problems that may come up and develop solutions proactively. I'm very concerned that as we start to see regulatory approvals and products coming to the market, one bad actor could set the entire industry back. We want to avoid that. We need to build public-private partnerships because this is really the only way that we can foster collaborations across all actors, especially industry developers who have the products that we need to use to develop these safety methods and data, and the research practitioners who have the knowledge and experience to be able to assess those, evaluate their safety, and answer these research questions. So the goal of this would be to develop and validate analytical methods, generate data sets that can be used by many stakeholders, and that would support transparent food risk assessment and policymaking processes across the world. And by making that data more open, it has a lot of benefits. I mean, I'll bring back again this idea that skepticism arises in the absence of independent data, and the only way to combat that is to have more transparency in this field. But it also accelerates the innovation and regulatory process, and we can take examples from other food innovations that have undergone processes like this. So, for example, there are databases out there food composition or genetically modified plants that can be used in this as an example as we try to develop these types of tools for this industry. So what is on the horizon? And this is my own crystal ball with a little bit of optimism thrown in. But I think we are going to see an increase in open research on safety. So we are finally starting to see government-funded institutes around research. And for example, the USDA National Institute for Cellular Agriculture, which is housed at Tufts University, does have food safety research included in its scope. So I think that will be a really great starting place for the academic space of this field. We need more public-private partnerships to address these questions, providing the samples that we need to generate the data. We're definitely going to see additional regulatory reviews. There are many in process, both in the US and across the world. And at least in the US, FDA documentation does provide some insight into the industry, which is really beneficial. And as this happens, as we see more regulatory reviews happen and products coming onto the market, we are going to absolutely see more hype and more criticism. But I'm really hopeful that we're going to also see more transparency because it's the first couple of points. So I'm hoping that can come back some of that hype and criticism. That's it for me. I'm happy to answer any questions, and as you're welcome to reach out.
Perfect. Thank you, Brianna. If you guys want to both go ahead and turn on your cameras, we can go ahead and get through the Q&A. There's quite a few questions here lined up in the chat box. So I'll start with you, Kantha. So you'd mentioned in your presentation that it would be unwise to extrapolate food safety rationales from animal-based to plant-based. Obviously, there's going to be different risk associated with each of those products. So what would be a more appropriate food safety rationale for these alternative proteins?
That's an excellent question. It actually requires us to go back to the basics. So when we looked at the safety of plant-based foods in the original sense of the word, as in just foods that are made with plant materials, we did not go into extraction and concentrations as we're going into it today. So to identify that our food product, our plant-based food product, is indeed safe, requires us to take into consideration the source of the protein, the process by which the protein was made, meaning it was a kill step of any kind, and then the type of processing that was involved in making the product, and whether the product is ready to eat or the product requires to be, needs to be prepared before consumption, so as to understand the journey that the microbial contaminants might go through, and then identifying, a model for each of the food product. What complicates and what further confounds this issue is that, let's just take one product, a plant-based hot dog or a plant-based burger. No two companies make the products using the same ingredients or the same processes. So even if you develop it for one, you can't really apply it for the other. So that is what causes it to be very complicated at the moment. I hope I answered your question.
Yeah, no, that tackled it. I know it's a big problem to tackle and probably a pretty open-ended question. So both of you touched on allergens, and there's quite a few questions here in the chat box about allergens present in these kind of proteins. How can you see how we can address the potential allergenicity of these products? I mean, there was a mention about education for consumers and clinicians, but how do you guys see the best way to tackle this allergy problem for these kind of proteins?
Dr. Duffy, you want to take it first?
I think that the education piece is really interesting to start off on, and that also ties with labeling of these products. So in cultured meat is especially challenging, I think, because on the markets, consumers are first used to plant-based alternatives. And so they're assuming it's a plant-based alternative. Whereas the allergens from the actual meat, so like, for example, a pork allergy, if it's a pork cultured meat product, consumers need to know there is pork in there. If you are allergic to pork, you will be allergic to this product. And so that's, on the one hand, where labeling becomes very important. And then you do have the variety of other ingredients that are going into there. some of which will be easy to try to avoid the common allergies when we're choosing ingredients for these products and labeling if those are included. But there is the potential that as we introduce new ingredients that have not historically been used in food, that new allergies could arise. And that's going to need to be something that regulators are looking at as these products go on the market.
And there's something else to add to that, and that is, remember, the science of allergens is a relatively new one. And in my presentation, I talked about grain millers developing an allergy. So these are people who have chosen the profession, and over time are finding out that they're becoming either sensitive to or actually allergic to that very same material that is part of the livelihood. So with allergens, as this category grows, and as these different ingredients become more prevalent in different food products, so will the exposure to these types of ingredients and these proteins. And only then will we find out which one of these are actually important ones or serious ones, and which ones are more, I would say, allergen causing than the other. And right now we are in that quadrant where we don't know what we don't know. We just know we have to be careful.
That's fair. That's fair. So Brianna, you talked about the microbiome of these cultured foods, and while maybe a benefit is perhaps they're cleaner, so it's a less diverse microbiome. Has there been any research to add back in potentially bacteria from beneficial standpoint, maybe perhaps to protect against those pathogenic bacteria that could possibly take up that niche? So has there been any work in that kind of field?
As far as I know, there has not been any research in that. And I would highly doubt any companies have pursued it yet, but it's absolutely an idea that's been floated. And I think first we need to, there's just so much research that needs to go into that. First, we need to understand what the population is going to be on these products, then look at how that's going to impact the potential colonization of pathogenic bacteria, and then look at potential colonization. But that is absolutely an idea that I've heard floating around the industry and love to see research on.
Bacteria doesn't always get the best rep, as they always get the negative connotation, but they actually have quite beneficial uses. So it would be nice if we could use them for good. So I know we're talking about food safety, but there's been a couple questions about the nutritional value of these products. Can we touch on a little bit of how they compare to traditional animal-based meats? There was a question specifically asking about cholesterol. So can we just touch briefly on the comparison between these alternative proteins and their cultured counterparts?
So we're going to take cholesterol, for example. We do know that what we know about cholesterol physiology today is somewhat mixed up and not very clear. So what consumers think that they would get cholesterol from, and how cholesterol really is formed in the body and shows up, to cause havoc, are two different things. So when you're talking about plant-based, I believe that we would need to first figure out the amount of a particular protein that is being consumed, but also what that formulation has. So the cholesterol implications of a product could change significantly depending on the type of fiber it contains and the amount of fiber. So at this time, to be able to make a comment about, are these products responsible for raising your cholesterol? Could they actually help you decrease your cholesterol? Your guess is as good as mine, because it is on a case-by-case basis.
Bree, did you want to add anything to that?
Yeah, well, I think with cultured products, it's still really quite early, where we don't know what these products are going to look like on that level. But I do think there are some exciting opportunities to tailor that a little bit. So you might be able to make these products healthier than conventional counterparts. But that's still yet to be seen.
Okay. When we're talking about trying to get these products into the market, we have a couple questions about scaling up the production. So how do we get these products to a scale where they can get into the modern market, or the big market, while maintaining its quality and safety?
I think that's for you.
Scale is such a big question in this space. As I said, the products that have seen regulatory approval and are quote unquote on the market are not even close to the scale that we would need for any sort of displacement of the market. So scale is an enormous problem that still really needs to be solved. I mean, companies claim to have solved some of these challenges on the scale internally, but there's no external data to validate that. So I think there's a lot of interesting discussion in the field right now as to where those blockers are currently and if and how we can get past them.
Okay. Kind of along that same line, so we're seeing a lot of startups, a lot of people trying to tackle this problem, come up with new products. But what extent are we seeing that conversion from traditional animal-based protein meat eaters to this now new alternative protein? Are we seeing a pretty big switch in the market?
I think there is a significant interest in these plant-based proteins. But in each category, it's taste and convenience that makes a difference. And of course, is it available? Because sometimes you like something and then you find that it's a startup and they haven't really scaled up, so you can't get the product, and it's affordable. But when you come back to it in terms of, are they consistently changing from the animal-based to its plant-based counterpart? Yes, if you look at what's happened to almond milk or soy milk, first it was soy milk and then almond milk at least surpassed soy milk in consumption. So that's good news. But when you look at all the plant-based analogs of whether it's cheese, dairy products, meat, poultry, etc., your guess is as good as mine. People want them. They want to want them. That's a very big, important thing. And they want to like them. Remember that these products, when they are demonstrated, are usually in the hands of an experienced chef or a culinary professional. But when you take them home and then you cook them, and you find that suddenly there are off notes and the rest of the family is not it's not warming up to the product, then going back and buying it a second time is a bit of a tall order.
Oh, that's a great point. So we're coming to the end of the questions here. I will wrap up and say maybe have a final question for both of you. I know Bree touched on it a little bit, but what do you guys see as the number one food safety issue moving forward that we need to address with these alternative proteins?
I believe the number one issue really is identifying the amount and the source. So identifying what are the major proteins in the marketplace. Are they all processed the same way so that the protein derivative are pretty much identical? So you may get the protein from pea, but the way you process it might result in 2 or 3 different types of proteins, and therefore 2 or 3 different types of allergen issues. So trying to map it and bringing them all to the same page so that you're comparing peas to peas and beans to beans would be the first step. Once that happens, then you'll have a little more confidence that we know what our ingredients are and where our allergen issues are stemming from.
Bree?
Yeah, I don't even know how I would pick just one. So, I mean, the list is quite long of things we don't know, the known unknowns, even not even getting into the unknown unknowns. But luckily, I think in almost every instance, there are clear next steps and places to take from other industries. So my answer isn't meant to be like, oh, there's a million things they're out of state. It really just is the work that needs to be done to, as I said, identify that list of all of the inputs so that we can even assess what do those residues look at the end, what is happening to us to go through. So I guess the first step, I think, would really be to gather that initial information and put that out there publicly so that work can be done.
Okay, great answer. I mean, yeah, that's a tough question and a pretty open-ended one, but no, great answers on both ends. All right, so as we're wrapping up, I just want to do a quick plug for the IAFNS Innovation Showcase. It is a virtual event scheduled for December 12th through the 14th. We are now accepting abstract submissions for the events, and you can find more details and registration information on the IAFNS website under upcoming events. And with that, I would like to give a huge thank you for everyone attending this webinar, and especially a huge thank you to Dr. Kantha Shelke and Dr. Bree Duffy for you guys both sharing your insights on alternative proteins. So go ahead and check out IAFNS.org, our website, for more information on upcoming events and ongoing projects. And make sure you give us a follow on Twitter and LinkedIn. So thanks, everyone. Have a great rest of your afternoon.
Thank you for your
Thank you, Caitlin.